Okay. We are going to get started for today. A couple of reminders here for you. Today we're going to be finishing our lecture on enzymes, so I don't want to rush through anything. So we do have probably a little over half the lecture left, so we're going to be focusing on that today. And then I do have a little bit of an activity for you at the end of class. So that's what we've got going on. Wednesday, I'm going to be talking about cellular respiration. So if that's a topic that causes you any amount of stress, hopefully it'll be okay. It can be fairly vocab heavy, but I try to break it down in a fairly reasonable way. Friday, I will not be here. I'm going to a science event with some folks in the department. So I will post a lecture on photosynthesis on Blackboard. It'll be a lecture recording, so watch that on your own time. It'll be in the folder lecture recording. So make sure you check that out. Again, we won't have class in here on Friday, so you don't have to come here. Do check out that lecture. And if you have questions as you're watching that, going through that, just let me know. Something that is a little bit challenging, of course, in having like pre-recorded lectures is that I don't have the opportunity to answer like questions as they come. And it's hard for me to see the looks on your faces to tell if you're understanding what I'm saying. So if you watch that and you have questions, just let me know and I am happy to address those. Questions here on any of this? Cool. So I did show you this slide. Last class. So I'm just going to put all this information back up here really quick. When we were talking about enzymes, remember we have a couple of important concepts. We have the idea of our substrate complex, which is where our substrate and our enzyme have bound together. The substrate is in that active site, and that's when the enzyme is actually able to catalyze the reaction. Remember, a key word here is substrate, which means the thing that the enzyme is working on. That can either be pieces that it's building together, things that it's breaking apart. And another key reminder here, each enzyme catalyzes only specific substrates. So later when I have you working on the little activity, you will kind of get an example of that. You'll be looking into a specific enzyme or a specific example of how enzymes work. Questions here? Remember this okay, hopefully? Cool. One key piece, and part of the reason that I'm showing you this again today, is to remind you about the active site, which is a really key location for this enzyme substrate complex. The active site is unique, and the active site is, again, formed during the protein. So when it's going through that primary, secondary, tertiary kind of shaping as a protein, the active site is actually formed during that process. So the shape of that active site is critical to the function of the enzyme. And that's important for what we're going to talk about here in a second. I did show you this slide as well previously. I just wanted to once again re-remind you, there's a lot of different ways that enzymes can lower the activation energy, which is the amount of energy needed for a reaction to occur. And enzymes can lower this in lots of different ways. We talked about potentially moving the molecules in an optimal orientation, maybe contributing to a specific environment, maybe actually donating pieces of the amino acids. So there's lots of different ways that this is going to occur potentially. I also mentioned this idea of kind of enzymes adjusting to be more dynamic in order to better fit with their substrates. They have this initial contact period, they have this kind of movement, and this is what we refer to as induced fit. So this idea of enzymes dancing is essentially the idea behind induced fit. And remember, I said it's like a firm handshake, basically like the enzyme is able to adjust to really hold tightly to that substrate in the most optimal way. So this is an active area of research. I personally find this super interesting. And if you want to read more about this, as always, just let me know. So now we're getting into new content. I've not shown you these slides before. So as I was mentioning, the 3D structure of proteins are super important. And actually, the three-dimensional structure of proteins are very subjective to their environment. Now, I have this bolded here because it's super important. Changing the environment of the enzyme can change its structure and therefore the active site. So kind of getting into what that means, if the enzyme is in a specific like cellular environment, for example, if there are changes to that, whether that's like temperature or pH, there can be alterations in that protein's structure. Now I have this bullet here, form equals function, which is referring to the idea that the shape of something is really important to the way that it basically operates. So I have a question here for you. There are changes to the active site. What does that mean for reactions that the enzyme would normally catalyze? And I want you to think about this. If you know the answer right away, that's okay. But I want you all to kind of think about this. If there's changes to the active site, what does that mean about the reaction? What does that mean about the substrate and the enzyme? So try to think about this, come up with an answer here. If what happens, so if there's like a change to the active site, so that specific location where the enzyme and the substrate interact, and it's a very specific shape. If there's changes to that location of the protein, what does that mean for the reaction that the enzyme is normally going to catalyze? Yeah, it can't catalyze it, or it might take longer. Both of those are true. Absolutely. Yep. So if there are changes to the active site or the specific location where the enzyme is acting on the substrate, most of the time the reaction won't occur because that shape really matters, or there can be delays or problems with that reaction. Does that make sense to everybody here? If there's changes to the active site, the enzyme won't be able to catalyze the reaction. The reaction may not occur. Now, something that can definitely impact this are changes to pH and temperature. Enzymes very often denature under high temperature or high pH. There's actually an optimal range of pH for each specific enzyme, essentially. Now, most of the time, the optimal range for a specific enzyme is fairly narrow, so they're not able to kind of work outside of that very narrow range. Now, when a protein loses its shape, we say that the protein has been denatured, and this is a key word here. The denatured refers to changes in the shape. So an enzyme has been denatured if it can no longer catalyze a reaction. If the shape of that protein is changed, maybe because of high heat, it can no longer catalyze reactions. Questions here? Okay. So I do have this slide showing some kind of weird figures here. In the top image, we have some pretty simplified representations of the enzyme here, and then this yellow blob is representing our substrate. Now what we're seeing here is the influence of high heat and high pH are causing this protein to change its shape, and we could see that after that high heat, high pH, the protein is not the same shape. The enzyme can't function the way it normally would. Same deal with that bottom figure as well. On the left side, we have a properly folded protein. And in this figure, we're seeing some of the spirals, which are representing those critical three-dimensional structures in the protein. Same deal here, the denaturation process because of high heat, high pH. Potentially, there's other conditions, which is what's being shown in the middle as well, like the plus and the minus signs are representing ions, so other sort of chemical or cellular changes can cause these effects as well. But again, this is just to highlight the specificity in shape of enzymes and how important that is. As I was mentioning previously, enzymes work within a specific range of pH and temperature. Typically outside of that range, the activity will either slow. So sometimes we say the rate of the reaction is going to be decreased. Sometimes it might not happen at all. Now, most enzymes in human organisms, for example, have an optimized pH of 7, which would be a roughly like neutral pH. However, if you think about some other conditions that exist within humans and other organisms as well, oftentimes the stomach, for example, is a highly acidic environment. So we see a really low pH in this type of organ. So I have a question for you. What do you think? How do enzymes function in this environment? If I said most human enzymes are optimized around a pH of 7, and then the pH of the stomach is 1.5, how do they do it? Any thoughts on that? They have different amino acids and maybe a different optimum pH. Yeah, that's a good thought. Does anyone have any other thoughts here? How is it possible? maybe reaching back to some of our ideas about like characteristics of life. Typically, the reason that we see this is because these are adaptations that have evolved over time. So the ability to be able to digest certain nutrients is advantageous, and we've seen this development over time. So here I'm showing some of these important digestive enzymes on the screen here, and these are associated with specific types of human food sources and different types of macromolecules. So the bread is representing carbohydrates. We have this like chicken leg thing representing proteins, and then our lipids down here with the oil. These are showing different specific enzymes, amylase, trypsin, et cetera, that digest specific nutrients. So that can be fun. I know students oftentimes have interest in kind of like human health or performance and things like that. There are specific enzymes for each type of food source that have to break those down. And then vice versa, there's specific enzymes that help build those certain macromolecules back up inside the body. So kind of interesting there. Now this graph is honestly, it's a little bit boring, but kind of a good visualization of what I'm talking about here. So this is showing the rate of reaction for two digestive enzymes over a range of pH values. So what we can see on that x-axis are pH values and then the rate of reaction. So the peak is going to be where the reaction is happening the most. This would be that optimal. So we can read this figure here at a pH of 2. Pepsin, which is a stomach or a digestive enzyme, is optimized at this pH. We see its peak here. And then we could look over here for trypsin. It has an optimized pH of 8. So that's just what this figure is kind of showing here. Questions on how I was reading this graph? Cool. So I have a fun little enzyme example. Hopefully you didn't read ahead. If you have not read ahead in the PowerPoint, good, don't do it. Science is about problem solving, so I'm going to set the scene here for you. We're going to pretend it's 1960, which was the era of Jell-O salad in lots of cookbooks and things. So you want to cook a delicious meal. You see a recipe in Better Homes and Gardens for a pineapple Jell-O salad. So you go to the store, you pick up a fresh pineapple, you pick up some Jell-O, and you're going to make the dessert. You're excited. This is going to be great. You combine the fresh pineapple, the Jell-O, you put it in the fridge for at least 12 hours, which would be a sufficient amount of time for Jell-O to kind of take its shape. However, you come back the next day to find the Jell-O has not set. It's a bowl of colored liquid and pineapple chunks in your pan. So the following week, you decide, okay, I really want to make this pineapple Jell-O salad. I really want to make this beautiful dessert. So you remake the recipe. This time when you go to the store, they don't have any fresh pineapple, so you get canned. After 12 hours, you follow the same procedure and you get this beautiful set dessert. So my question is, what gives? What the heck happened? I want you to think about this with a partner. Try to solve this mystery. Do your best, come up with a hypothesis. And I don't want you to use the internet. You could easily Google this. I don't want you to do that. I want you to use your brain. Think about what we've been talking about here today. Think about what you've learned about enzymes. And I gave you a hint. What are the differences in the way the salad was prepared? Why might that matter? So I want you to think about this with a partner. And if you already know the answer, don't yell it. I want you to think about it and talk about it with someone near you. Yeah, come up with a hypothesis for this observation. We're practicing the scientific method here. What do you think?
Getting something to do with the enzymes in natural pineapple that would not be in the canned pineapple due to preservatives or something.
The fresh pineapple versus the canned due to preservatives. Okay, interesting. Good thought. Anyone else? What do y'all think back here? What's going on?
You said canned pineapples have more enzymes than fresh pineapples.
Canned, say that one more time.
Yeah, or maybe the fresh pineapples have more like active enzymes than the
They have
canned
more
ones.
active enzymes. That's a great thought. What are you thinking over here?
So the enzymes in the canned pineapple is already denatured, so it cannot react with the jelly.
I was thinking the same thing.
Can't react because they've been denatured. Okay, good thought. Active versus inactive enzymes, maybe.
Like more or less.
Okay. Good. You haven't scrolled down yet, hopefully. No scrolling. I shouldn't have posted the answer. Cool. What do y'all think? Ooh, that's a really good thought. The aluminum in the can taking away like the acidic conditions. That's really interesting. Good thought. Mm-hmm. Mm-hmm. You have a good hypothesis? Hey, lay it on me.
Like maybe their power went out.
The fridge wasn't on.
The fridge wasn't on. Honestly, that's a really good hypothesis. Yeah, maybe the fridge turned off.
Confounding there.
There's too many confounding variables. Yeah, that's kind of funny. Okay, so that's a good thought. I mean, when we talk about the scientific method, a lot of times we assume it can only be applied to really like scientific problems or problems that are easy to test. But kind of the idea behind the scientific method sometimes can also be like, okay, let's say you have a flashlight, right? You go to turn the flashlight on and it doesn't turn on. You're kind of using the scientific method if you check like the light bulb, if you check the batteries, if you have a hypothesis, okay, I think the batteries are dead. Put fresh batteries in, the flashlight works. That's kind of like a simplified version of the scientific method. So that's a good thought here. One hypothesis, maybe the fridge was off. Yeah, that's not what I'm getting at here, but that is funny, a good idea. I heard some others, maybe the aluminum is changing the acidity. Okay, very creative there. I heard some others, maybe we have more active enzymes. Maybe there's something to do with the enzyme shape. And I will say lots of you are very close. So something about fresh pineapple, it has the enzyme bromelain, and bromelain is actually a protein digesting enzyme. So what is Jell-O made out of? For one, sugar and also food dye and flavoring, but it has collagen and it has gelatin, which are animal proteins, and that's what causes Jell-O to set up and become firm. That's what Jell-O is actually made out of. So we know that gelatin is an animal protein derivative, and as a result, this bromelain enzyme actually digests the proteins in Jell-O that cause it to set up. Now, this is true. This isn't made up. If you really tried to make a Jell-O salad with fresh pineapple, it wouldn't work because those enzymes in the fresh pineapple actually do digest the protein in the Jell-O that causes it to set. Why does the canning process fix this? The syrup, that's a good thought. So canned pineapple actually doesn't have syrup in it at all. It's just a fresh pineapple. But in the canning process, has anyone ever canned something at your house? What do you have to do? Yep, and what do you put it in? A jar, and then the jar goes into what? A pot of boiling water. Yeah, absolutely. So part of the canning process, now with industrial canning, things get heated and cooked. So it actually gets heated up really high, which is how those things, whatever's inside the can, when I say those things, I mean whatever product is inside the can, essentially gets cooked. Same deal with tuna. They put tuna in the can fresh or raw, then they heat it up in the canning process, and that's what cooks it. So same deal with protein, with pineapple here. We heat the can up really hot, and then that denatures this bromelain protein. Wow, crazy. So 250 degrees Fahrenheit is actually the canning temperature, so they do get it really, really hot. So questions here? Kind of a fun example. If you want to try this out at your home, you can. Jell-O salad's honestly pretty good. Okay, a little bit more detail here into enzymes. Sometimes enzymes need additional components or molecules, things like that, to actually work effectively. We have two main categories of these like helper components. We have cofactors, which are inorganic molecules. I have some classic cofactors listed here for you: zinc, iron, and magnesium. Now, remind me, what does inorganic mean? Yeah, typically we don't have those carbon bonds. Yep. Or we don't have carbon-hydrogen bonded together. Yep, absolutely. And then we can have coenzymes, which are organic molecules. A lot of times these are dietary vitamins. Now, sometimes cofactors and coenzymes are going to bind really tightly and permanently to an enzyme. Other times they're going to be loosely bound and then they'll later detach. But essentially, cofactors and coenzymes help enzymes catalyze reactions. So if you're the type of enzyme that has to have a cofactor in order to work, without it, you won't be able to work. That's the basic idea here. So sometimes enzymes can only catalyze a reaction if they have these specific components. Now, a lot of times these would be supplements. So if you went to like, I don't know, online, if you Googled like important supplements, a lot of these would pull up. Zinc, iron, magnesium, those are all really critical types of molecules that we need to eat or types of elements. Most of the time you would get these from your diet, both cofactors and coenzymes. Now I have some examples of applications for these. DNA polymerase is a specific enzyme that helps build DNA. We're absolutely going to talk about DNA polymerase. I have a little picture of it because it's super duper important. So DNA polymerase requires zinc in order to build DNA. Vitamin C is necessary for collagen production. Collagen is an important protein for connective tissue. We also know sometimes cofactors and coenzymes help to regulate enzymatic activity. So when I say regulate, that means either promote activity, so make it happen more, or kind of keep it from happening as much. So they can either promote it or kind of inhibit it. And as I was mentioning on the previous slide, typically organisms get cofactors and coenzymes from their diets. And I have a picture of the kiwi, the lemon, whatever else is in that slide, limes, grapefruits, things like that, because those are good sources of vitamin C. Okay, I do have a break here for you. I'm going to bring around little pieces of paper. This is your break. What is your favorite song right now? No gatekeeping. And I want you to write it down here. And I also want you to share with someone you don't usually talk to. Yeah, pen or pencil. Just write it down so that I can know. And then I want you to share with some folks. I think you need one more. You have 3? Sweet.
Well, repeat on Spotify.
Yeah, see what you've been listening to the most. Do you need more back here? Cool. Everybody really mixed up where they're sitting today. This is so interesting. It's hard for me to get apart. It's definitely not. Do you need another one? What's my favorite song right now? No, this is just what I was listening to when I made this. Do you have enough little cards?
I think so. We have more than that. Favorite song.
What's my favorite song? That's honestly a hard question. I'm gatekeeping. I really like Sally by Role Model, but that's a pretty cliche answer, I feel like right now. It's a good one. You should listen to it. Sally, When the Wine Runs Out. I honestly listen to a lot of country music. Listen to a lot of like folk. Well, I said right now, I think it's probably Sally When the Wine Runs Out by Role Model, but that's kind of a cliche answer. Yeah, and it doesn't have to be your all-time favorite song, just your favorite song right now. And then tell a friend. "49 Winchester is great though. And then make sure you give me this before you leave so then I can listen to all your songs. Actually I do, yeah. I usually do. I don't listen to ones I know, but yeah.
You like this one.
Sometimes I don't. I'm like, whoa, that's a rogue choice. And sometimes I'm like, great choice.
You have to turn your volume down.
I'm just in my office, like really loud.
Some songs are a lot louder.
Oh, some that are like aggressive. I have headphones. If you have a favorite band, try to choose your favorite song.
Wait, no, I got it.
Okay, good. And then make sure you share with your friends. Kristen, share with your friends. Share with Celine. You shared with Rainier? Okay, cool. Great. Yeah, and make sure to give this to me before you leave.
Which one?
Think cloud music.
Clown music.
Like,
Oh my gosh.
cloud.
Electro clown.
Oh.
Loud. Okay, I thought you said.
Loud music.
I was thinking like, are you from small town?
No, I'm from like Boulder, Arvada. Yeah, a little bit. Fair bit. I'm trying to think of what else. I honestly recently have been listening to a lot of audiobooks.
Audiobook.
My favorite, I listen to a lot of like, it's kind of weird. I'm training for a half marathon and I'll like go on long runs to like nonfiction audiobooks. Kind of weird. Cool. Yeah. Lovely. Lovely. Woohoo. Yeah, you can pile them all up maybe. I'll come grab that one in a sec. Ready? Yeah, you can pile them up. Then it'll be quicker to pick up. Boom, boom. Ah! Great. This keeps me inspired.
Good.
Thank you.
Like this might be awesome.
Pre-games. One of my undergrads, whenever she gives research talks, she's like, I need my pre-game song. I need my walkout.
Like, go.
Awesome. Thank you. Duran. Woohoo. Where did I put my glasses? Ready? If I didn't grab these, you can bring them to me later.
Do you ever listen to them?
I do. Yeah, I always listen to them.
Everyone?
Pretty much, yeah, I go through them all.
They're like immediately skip.
No, not usually. I mean, sometimes I won't listen to the whole song. Sometimes I'm like, I've heard enough. But most of the time I do listen to all of them. Okay, everybody have time to share with a friend. Or the partner, yes. You take yours too. I think I got pretty much everyone else. 49 Winchester's great. Okay, we're gonna keep talking about enzymes here just a little bit. We have about 25 more minutes. So something I also wanted to mention, because this is really critical relative to kind of like applying these concepts to real world situations. A lot of times regulation is super critical to kind of how enzymes function and their actual functioning in living organisms. So I have this kind of dramatic title: Chemical Chaos is Subdued by Enzyme Regulation. As I was mentioning, cells have lots of different enzymes, and we don't necessarily want them working all at once at the same time. So for any one molecule, there's often an enzyme that breaks it down and builds it back up. Now you can imagine what might happen if both the building and the breaking enzyme were active at the same time. What would be happening? Yeah, they would cancel each other out. They would be counteracting each other. Basically, we would have this opposite, these opposite enzymes working at the same time. All that they would be accomplishing is using up cellular energy. Now, organisms typically don't do things that just waste a bunch of energy. So cells have a preventative measure for this, and it's through enzyme regulation. Now, there's a couple of different types we're going to talk about. We have competitive inhibition and allosteric regulation. So these are our two main categories for how enzymes are regulated. And I have figures on the next couple slides. So more on these in a sec. Now, competitive inhibition, when we break that kind of word combination down, competitive means like competing, of course, and inhibition means to block, essentially. So with competitive inhibition, an inhibitor, which we can see in this figure, is like this little blue molecule. An inhibitor is a molecule that can fit directly into the active site and block the enzyme's ability to interact with the proper substrate. So competitive inhibition, we have a molecule blocking the substrate's access to the active site. Whereas with allosteric regulation, we have a molecule binding to the outside of an enzyme that either speeds up or decreases the enzymatic activity. So I have a picture for this on the following slide. So competitive inhibition, a molecule matches in at the active site. Allosteric regulation, we have a molecule binding to the outside of the enzyme. Now, as I was just kind of saying, sometimes with allosteric regulation, we can either have inhibition or activation. So that molecule binding to the outside of the enzyme can either speed the process up, which is what happens when you have allosteric activation. This little orange square is representing an activator molecule, which is going to make this happen more. The reaction is going to happen faster, and more of that product is going to be produced. Whereas with inhibition, same deal here as with the previous type of inhibition I was talking about. It gets slowed down, but the way that it gets slowed down is just because there's an interaction from the outside. Now, the word allosteric actually refers to a location separate from the active site. That's kind of what that word means. Questions here on enzyme regulation? There's a couple of important real world connections to enzymes. Again, trying to emphasize this idea here. And I am going to have you all choose one of these 3 bullets and kind of look more deeply into the details of this. So I am going to have you come back to this, but some classic examples of how enzymes impact our lives. Penicillin and vancomycin are broad spectrum antibiotics, which means they're antibiotics that can work on a lot of different bacteria. They're medicines that can kill lots of different bacteria. Typically, the way that they do that is through enzyme inhibition, and they keep the bacteria from growing their cell walls. So if you choose that example, that's an important thing to know. A lot of insecticides and pesticides actually use enzyme inhibition to kill their target prey species. This is a little bit, I think, kind of interesting. Typically, the way this occurs is through blocking the enzymes that help with reuptake for certain neurotransmitters. Neurotransmitters are molecules in your brain that kind of help accomplish certain goals based on what the neurotransmitter is. Now, what some of these pesticides or insecticides do is they block the reuptake, and then you see lots of firing, and the brain basically has issues associated with that. So I'm not going to tell you more on this. If you choose to look into this one, it's very interesting. And then finally, if you've ever taken a prescription medicine, a lot of times on the bottle, it says like, do not drink while taking this medication. Most of the time, that's because of alcohol being metabolized by enzymes and the fact that some medications or drugs can actually impact that metabolism process because of these enzyme regulation properties. So enzymes actually do matter a lot in our daily lives. And I'm going to have, again, you and a partner probably look into one of these. Now, to answer the question I asked at the beginning of last class, why can't we go graze some grass right now? We don't have the enzymes required to break down cellulose. Now, remind me, what is cellulose? Yeah, it's a polysaccharide. Cellulose is really a very prevalent polysaccharide in plants. So we could chew up grass, maybe we'd get some fiber, maybe we'd have some slight digestive benefit from that sense, but ultimately we don't have the capacity to digest grass. So yeah, Would it be warm?
Corn.
Oh, would it be like corn? Yeah, it's kind of related to that. So like the sugary part of the inside of corn you can digest, but yeah, like the outsides you don't have the capacity to digest because it's made out of insoluble components. So what's up with cows? Do they have cellulase, the digestive enzyme for grass? Some people say yes, some people say no. They have to digest it in 4 different chambers. True. Yes, ruminants have a lot going on with their digestive systems. They don't have cellulase, though, which is kind of interesting. So they're able to harvest the sugars from plants and from grasses and things. As you were saying, they have lots of stomachs. These stomachs tend to be really conducive microenvironments for small organisms. So they're moist, they're hospitable, and they actually harbor microorganisms like bacteria and fungi that have cellulase. So they basically house other things that can digest cellulose. And then the cellulose is broken down into fatty acids, and that's how the cows get energy from it.
They form those bacteria and fungi in the-
Oh my gosh, that's a great question that I do not know the answer to. Are they born with them? I'll have to look that up. I don't know. It's a good question. Maybe when they're drinking it. Yeah, that's a pretty typical, like most organisms have to acquire their kind of microbiome. So like babies are really susceptible to diseases because they haven't encountered them yet. So yeah, possibly from a nursing process. Yeah, do you have the answer? Are we like this with dairy products? That's a great question. So human capacity to digest lactose sugars, the enzyme lactase, is related to that. It's actually more of like a genetic element for us. It tends to be like if your parents are lactose intolerant, you may be. It was actually a mutation. Humans were originally not able to digest lactose sugar, but in nomadic colonies, there were mothers and offspring, like babies, that were living longer when they were able to take in the calories and the fat of like dairy products. And so it became advantageous. And then we saw it kind of more widely spread in the human population. But yeah, it started in kind of nomadic, I think it was with goat herding tribes. But that's a great question. Lactase is an important enzyme as well. If you don't have lactase, you know how important it can be if you like ice cream. Any other questions here? Cool. Okay, so this is what we're going to spend the last like 15 or so minutes on. And I do have more paper for you. I have little half sheets that I'll bring around. I want you to use one of the real world examples I listed on the slide. So either antibiotics, insecticides, pesticides, or drug alcohol interactions. And I want you to kind of look into one of those specific examples. I want you to illustrate enzymatically what's going on. And I want you to find the name of the enzyme and label it. You can work alone, you can work with a partner. I do not care. I do recommend you put your name on this. I think I'll probably give you 2 points for it. If you'd like, if you don't finish it, I cannot give you points. I don't know. I think I'm going to give this 2 points. So choose one of these examples: antibiotics, pesticides, drug alcohol interactions. If you need help, let me know. I can help you get started for kind of what to look up or what to investigate. You get to choose. So paper for you here. And if we need more, I guess I can go find more. You can rip these in half. So you can work with a partner, you can work alone, whatever you like.
Which one do you think we should do?
I personally, what are you most interested in? I feel like the antibiotics are the most boring to me personally. The drug alcohol interaction is really interesting, absolutely. Pesticides are kind of crazy. If you scroll down a little bit more, keep going, a little bit more, right back, keep going. Sorry, keep going. There you go. Yeah, you learned a lot about enzymes. I don't know how many pieces of paper you need. Yeah, if you want to work together as the 3 of you, that's fine. If you want to do whatever, I could take those if you're ready. And if you need help finding the name of the enzyme you're looking for, let me know. If you need help with some vision of where to kind of start your research, let me know. And you can work alone, you can work with someone. Do you want a piece of paper? Cool, yeah. Yep. Does everybody have paper who needs paper? Cool. And like I said, put your name on it and I'll give you 2 points for this.